Actinomycetes, and I had the pleasure of knowing him 25 years ago, and since then I have been one of his admirers. So Dr. Goodfellow, you have the podium.
Okay, I would like to start off by thanking Professor Fahal and his colleagues for inviting me here. It's been my pleasure to come here to Khartoum, as some of you know, several times, and I'm really pleased to see the Mycetoma Center gaining in status year in, year out. And I think, rather like Paul said before, those of us who are bacteriologists, we feel really rather humble when we consider all of the fine work that you're doing here, and we're just people that are rather interested in the organisms. So I'd like just to underline the tribute that we should be paying to Professor Fahal and his colleagues. In my view, people like him are heroes of our time. But he has asked me to talk about actinomycetoma new causal agents, and this is what I'm going to do. And I want to try and keep it very simple, because I'm not wanting to talk to my microbiological colleagues particularly, but I'd like to talk to those of you who are not microbiologists, because I do sense that, and I can understand this, that what we do as microbiologists is rather secondary when you're faced with the practical problem of treating mycetoma patients. But nevertheless, I think the microbiologists have a role to play. So my main themes are outlined here. I want to talk about the ground rules for describing new bacterial species. This will be familiar to some of you, but really the point that I want to make is that I think this sort of work, without too much trouble, can be done here, and I think it should be done here in the near future. So I want to go through some of the more schematic the concepts underlying classification and identification, particularly classification. And then I want to say something about the actinomycetes with a particular objective in mind, and then I want to talk about some of the causal agents, and I want to convince you from what I say at the beginning that this work's been done properly and that the causal agents that we're going to introduce to you are different from the ones we know. And then towards the end I'd like to talk about outcomes and directions of travel. The work that I'm going to go through is really a tale of three cities, Khartoum, Newcastle and Wrocław. This is where most of the work's done. And I now see Paul in front of me, and it should really be 4 cities, because we should have Liverpool as the number 4, because my interest and work on actinomycetes and organisms that cause mycetoma started way back almost 50 years ago. So very quickly, what's prokaryotic systematics? As you can see here, it's the study of the kinds and diversities of prokaryotes and relationships between them. And it includes classification, nomenclature and identification, but also rather a lot more. And some of the things that Paul said, some of the rather interesting things that Paul said this morning, would it come under the umbrella of systematics? And people sometimes forget about that, but it is important. You can study systematics from a theoretical point of view or from a practical point of view. my interest has always been on the practical point of view, because I don't think I'm clever enough to do it from the theoretical point of view, because that really gets you into quite complex mathematics. But I also think that people like me should be trying to help people dealing with real problems like you've got here. So we recognize these three divisions of systematics. They should be done in this order, and I spend quite a bit of my time trying to explain to people that really classification and identification are very different. You first classify, then identify, and we tend to sort of use these words as if they're synonymous, and I think it's very important to use the terms in the proper way. So classification, as you can see there, we can use all sorts of techniques for assigning organisms to groups. The important thing, once we've classified, is we can give names to organisms, and there are international rules for doing that, so that once an organism is named, it really is a means of communication. We should take those names seriously. And then latterly, but certainly not last, we've got identification. So people like me always feel a bit embarrassed. I'm a taxonomist, I'm a systematist. People think we're not very useful, rather like the mycetoma agents. We're a neglected group. But we are very important for the reasons that are outlined in this slide. And I won't go through all of these. But what I do want to underpin is maybe three things. One is that classification gives us the scaffold on which to fit on the nomenclature. It's the very basis of what we do in microbiology. And there's a number of things that are really important, but from our point of view, today it's really the detection of new and emerging pathogens, and also the choice of strains for pathogenicity, whole genome sequencing, and biotechnology. If you choose the wrong strains, you can waste an awful lot of time doing work that it turns out to be valueless, and I could give you many examples of that, but I won't do that. So this is contemporary systematics really in one slide, so I'm not actually taxing you too much with systematics. Now the approach is what we term an integrated approach, using the genotypic and phenotypic data, as you can see here. And over the last 30 years or so, there's been three approaches. Chemotaxonomy, which is simply using chemical markers for classifying and identifying microorganisms. Molecular systematics, which Paul gave some very nice illustrations of the importance of that this morning. Molecular systematics is considered to be extremely sexy. Chemotaxonomy is considered to be dull. Chemotaxonomy is important and will remain important in my view. And I think when we look at the reclassification of Nocardias and Actinomaduras, it was actually the chemotaxonomists that got things sorted out before the molecular biologists came along. It makes me feel a lot better saying that, but the molecular biologists are going to have a good future. And then I'm an old numerical taxonomist. I applied statistical techniques for sorting organisms into groups based upon phenotypic properties, and I'm probably one of the first numerical taxonomists, and I think perhaps the last. But I don't think so. I think numerical taxonomy is going to become very important in years to come, but we'll be analyzing data coming from molecular systematics in the main, but not exclusively. So the outcomes of all of this, as far as bacteriologists are concerned, is we've got pretty good classifications now of the organisms we know, and less good identification systems. Here's our famous ribosomal RNA. I put this I flagged this up because the whole of the scaffold for the classification of actinomycetes and all the other bacteria for that matter, and archaea for that matter, is based upon the sequencing of 16S RNA. And you can see that we've also got 5S and we've got 23S RNA. The 16S was chosen because it's just about the right size. It's about 1,500 nucleotides. The 23S never really took off. It involved a lot of sequencing and some considerable difficulties with primers. The 5S was too low. This is the system that everyone uses now to derive phylogenetically based trees, and I'm not going to go into all of this. I just want to sort of flag this up. I think several people here in Khartoum are very familiar with this. They can do this, and I've put the slide up just to try and emphasize that this is something that can be done here locally once you've got the equipment, which is not terribly expensive, but if you don't have it, it is a problem. And the costs of actually doing the sequencing are fairly modest. So I wanted to flag up 16S RNA gene sequencing because while it's got some disadvantages, which I'm not going to go into in any detail, it has actually completely changed the classification of all prokaryotes. And it's a relatively cheap technique, as I've said, but the critical thing is that you can build up databases. And I did mean to check before I came how many sequences are in the 16S RNA database now, but I think it's well over 100,000. So this is marvelous. You've got a really good database, a reasonably good database there. People get a bit sniffy with 16S RNA sometimes, and they look for faults in it. There are faults in it. If the sequences are not good, you're distorting the relationships between the organisms that have those poor sequences. But by and large, I would really like to emphasize this has been extremely useful and will be useful for some years to come yet. And also we can look at the sequences and we can find nuclear signatures that actually define particularly higher groups of actinomycetes, and we can derive primers and probes for identification for organisms that contain unique bits of DNA in the 16S. There are some disadvantages. I don't really want to go into the disadvantages too much, because for most of the organisms we're dealing with, we can separate new organisms out with 16S RNA sequencing, with the possible exception of the Streptomyces. And they don't form a tree when we look at a lot of the sequences. They really form a bush, and that can cause us problems, which I'll return to it a little bit later. Paul talked about resolution a little bit earlier on, and the value of MLST, which is particularly useful at the species and subspecific level. The gold standard for resolution vis-a-vis 16S RNA sequencing is concerned is still DNA-DNA pairing. And maybe we should go through this slide a little bit carefully. So the extent of binding between DNA from a test strain and a labeled strain from a reference strain is expressed as the percentage relative to the extent of binding between the labeled and unlabeled DNA from the reference strain. So in other words, we've got a homologous reaction and we've got a heterologous reaction. And if in the heterologous reaction you've got a relationship that gives you a figure of 70% or over, those organisms are considered to belong to the same species. And that's really still, despite all of the fine molecular biology that's done, it is still the gold standard for defining species. I hope it won't be the gold standard for too much longer, but nothing has come along that gives us this level of resolution. And I would like to show you this slide, because this is expressing the 16S RNA sequencing values along the vertical axis, and along the horizontal axis we've got the DNA-DNA pairing. This diagram, in my view, is really extremely important for understanding contemporary prokaryotic systematics, because if you look at this plot, it's only the organisms in your top left-hand corner that are belonging to the same species. So if you get if in your 16S RNA sequencing, you get a sequence between an unknown strain and a known strain of less than about 98.7, you can be sure if it's less than 98.7, you shouldn't really have to do DNA-DNA pairing unless your paper is refereed by somebody that doesn't understand systematics, and there are many of those around, sadly, these days. I've been asked to do DNA-DNA pairing between organisms that have as little as 96% 16S RNA sequence in common, and this is just absolutely ludicrous. So there's a little bit of a battle there between people that understand the system and people that don't. I think the problem is that there's no linear correlation between the 16S RNA sequencing and DNA-DNA pairing. There's a particular problem with Streptomyces. Streptomyces form a bush. Many of these Streptomyces have 16S RNA sequencing well above 99%, and you really then have to do DNA-DNA pairing. Very quickly, what are Actinobacteria? I suspect most of us know that, high GC ratio organisms. They've got a few indels that allow you to separate them from other prokaryotes, but it's the bit at the bottom that I really wish to draw to your attention, and that is that until very recently, with very few exceptions, most actinomycetes were considered to be saprophytes and members of the soil ecosystem. We're beginning to find now that this is a distortion, and it's a distortion that's built upon the previous interests of people. For instance, if we take Streptomyces as an example, most people have been interested in Streptomyces because they're a source of new antibiotics. And to be quite frank, the people that were working with Streptomyces didn't want to have any of those very nasty pathogens from the Sudan causing them trouble with health and safety. So there was a feeling that Streptomyces weren't pathogens, or if somebody pointed out there were, well, the person pointing that out was wrong, they shouldn't be pathogens. And that's really one of the small points, but important points I want to address today. Here's I was talking about resolution before. You can see the techniques that we use to express relationships at different levels in the taxonomic hierarchy. 16S RNA sequencing is pretty useful above the genus level. A lot of people say it's not so useful within the genus. It depends what the genus is. Sometimes actually it is very useful. And then we've got techniques like Paul was talking about this morning, which give us a lot of resolution, and that's obviously going to be of extreme importance for epidemiological studies. So you choose your techniques depending on what you're trying to achieve. This is where we are with the phylum Actinobacteria at the moment. We've got 5 classes, as you can see, a number of orders, a number of families, a number of genera, and a number of species. And I've taken these figures from the volume 5 of Berger's manual, which is coming out later this year. I've, together with a few others, have been half killed trying to write the chapters for this particular volume, but that's the hierarchy we've got at the moment, and this is a little bit important to something that I want to point out in a moment or two. But first, what's the extent of actinobacterial diversity? If you look at molecular ecological approaches, you find that within 1 gram, let's say, of sediment in the sea, you might find over 1,000 different organisms when you look at the various clones and put them into the 16S RNA tree. We've done this in this work with GemStash, and we found that nearly all of these clones belong to different species. So the general view now is that we haven't isolated more than about 0.5%, 1% of the total variation that exists amongst the prokaryotes. So if you look at the work that's been done on cultivatable organisms, that's right at the bottom, we've got relatively few, I think, just with the bacteria as a whole, we've got about 70, got to get this right, about 7,000 species, I think. It's minimal. And this brings me to this slide, and it's a very simple slide, but I think it tells us really something very important. And that is, if you look at Mycobacterium and Nocardia in particular, you can see the rapid increase in the number of species that have been described over the last few years. And the figures in brackets indicate the number of organisms that are pathogenic. So if you look at Mycobacterium, we've now got 126 species. Over half of those are actually for organisms that are pathogenic. And it's a similar state with Nocardia. So the point that I really want to make is that Mycobacterium and Nocardia, particularly Mycobacterium, receive a lot of attention, mainly because people are interested in the Mycobacterium tuberculosis and Mycobacterium bovis and Kansasii, a few that are well-known organisms. The other organisms, incidentally, until recently used to be called the atypical Mycobacteria because they were thought to be of very little value. Well, we now know that that is not the case. If we look at some of the other organisms on this slide, the Actinoplanes, Streptosporangium, there's not been an increased number of species, and this is mainly because these organisms are quite difficult to isolate, but also because they're probably saprophytes. So people working in medicine they often don't like taxonomists because they say taxonomists keep on changing the names. But in actual fact, people in the medical profession are very good at taking on board new advances in systematics. And systematics, like any other discipline, is advancing all the time, and Paul gave us a nice indication of that this morning. So I'll show this slide really to emphasize the fact that if you're looking at organisms that are causing actinomycetoma, a neglected disease, as we've heard several times, we should be anticipating that there's going to be many new organisms that can be described that are causing this complaint. And that's why I show this particular slide. This is the classification. This is the sort of thing that drives non-taxonomists mad. Here I am showing you the hierarchic classification of the organisms that actually cause actinomycetoma. The Nocardias, as you can see there, and the two Streptomyces species, and then the Actinomaduras. Just a word about Nocardia that cropped up this morning. Nocardia is my first love. I've been working on Nocardias for many decades. In actual fact, in Thailand and in other countries that are under the tutelage of Japan, people in clinical laboratories take pathogenic Nocardias very seriously, and of the 36 or so new species that have been described, most of them come from that part of the world. The most significant species, however, causing nocardiosis, other than the well-known ones, is Nocardia africana, and that was work that we did here with colleagues in Khartoum. So that doesn't get the recognition it deserves. The point that I want to make here is made in the next 3 slides. So this diagram is based upon differences in 16S RNA sequences. These are our 5 classes. There's actually a 6th class here because one's just been added. And all of our pathogenic organisms are in the top wedge, the Actinobacteria, which is showing a tremendous amount of variation. The depth of that wedge illustrates variation on the basis of 16S RNA sequencing. Here we've got the mycolic acid containers that we've mentioned earlier this morning. And one of the things that intrigues me, that as agents of actinomycetoma, it's only members of the genus Nocardia here. So far, not any other members that contain these mycolic acids. And I find that a little bit odd, but I offer that just as a thought for you, especially since if we look at the what have we got here? The Streptomyces, they're in a totally different group. And then if we look at the bottom, I've gone through that slide maybe too quickly. If we look at the Actinomaduras, they're in a very different group again. So I haven't looked at the number of 16S RNA sequence differences between the Nocardias, the Actinomaduras, and the Streptomyces. I should have done that before I came here. But I reckon there will be 50 or 60 differences, which rather suggests that these organisms parted from a common ancestor maybe 30, 40, 50 million years ago. So I just find it odd that we get these mycetoma-causing agents in these disparate genera. And that's what makes me feel that there might be pathogenicity islands involved, and we'll come back to that at a little bit later. We've heard a lot about the Mycetoma Belt. I don't want to go into that. I think everyone here knows what's on this slide. The only point that I would make, and it's a point that Paul touched upon, is that I'm not certain that the Nocardia brasiliensis, for instance, that are isolated in Africa, on the African continent, will be the same as those isolated in Mexico. And same for Actinomadura, and same for Streptomyces. So I think we've got to be very careful with interpreting the labels we give to these organisms in these different parts of the world. And certainly with Nocardia, a point I tried to make the other day, if the classification's been done more than about 10 years or so ago, I don't think you can rely on that classification, especially if organisms are lumped together as Nocardia asteroides. With Nocardia brasiliensis, I've got a more open mind. So why isolate and classify the causal agents of actinomycetoma? Well, I'm pointing this out on this particular slide. Paul's touched upon some of this, so I don't want to go into it in too much detail. I would just like to point out at the end that if we really want to understand the epidemiology of the organism, we really have to devise new selective media for trying to isolate these pathogens. And I think we do have data on which to tackle this problem, and I think that's one of the areas that we should be giving some thought to. The other ones are quite obvious, of course. We don't really know in the Sudan which are the particular types of Streptomyces that are causing actinomycetoma, hence the need to look at this in some detail. So if we're wanting to classify agents of actinomycetoma, it's very much like classifying any other prokaryote, really. We've got to have the pure culture. If we've got lots of cultures, we've got to sort of dereplicate them, assign them to groups, choose representatives for more detailed studies. But the first port of call, if you think you've got something new now, is really 16S RNA sequencing. We do that routinely. I don't work very much with pathogens, but I do work with organisms, that actinomycetes, where we're looking for new organisms to produce new drugs. Every organism that gives us positives in the assays, we now routinely do 16S RNA sequencing, and nearly everything turns out to be new. With Streptomyces, however, you sometimes have to do this DNA-DNA pairing, as you can see down on the left hand of this slide. So that's the strategy, and it's a relatively simple one. So a little bit about Streptomyces. I think how we define Streptomyces reflects our interests. Paul had quite a nice definition of them earlier today. I've given you the taxonomic definition, and this is really meant to be the criteria that are particularly important to pin down that an unknown organism belongs to the genus Streptomyces. The quickest way is 16S RNA sequencing, but you can also do some chemotaxonomy, particularly looking for the marker L-diaminopimelic acid. The point that I want to make right at the bottom is that there are 553 described species of Streptomyces, most of these produce interesting secondary metabolites. So the whole classification of this genus has been skewed by people interested in looking for new organisms, new Streptomyces that might produce something valuable. And it's only relatively recently that people have begun to appreciate that Streptomyces are not just pathogens. Well, here in this part of the world you've known about this because of the organism called Streptomyces somaliensis. It's rather interesting that Paul may remember at the meeting we had in Newcastle where all of the Actinomycete world got together. I think it came as a surprise to them when in fact I gave a paper pointing out that Streptomyces really were serious pathogens. And I think we're back to the argument then, this is just going back 4 years, that people working on these organisms from other points of view didn't really want to take too much interest in pathogenic Streptomyces. I think that has changed now, and I think it's changed because of the work that's been done here in collaboration with people like myself. So I think people here in the Sudan should take a bow that we've got Streptomyces on the stage as being an agent, a human pathogen. A lot of work has been done in recent years on Streptomyces scabies. This is an extremely nasty pathogen of commercially significant potatoes, vegetables generally. Until very recently, there was one species recognized, Streptomyces scabies. We now recognize another 7 species. So this is what I anticipate will happen with or is happening with the pathogenic Streptomyces. So here we are again. If we've got an unknown Streptomycete, what do? Or we think we've got an unknown Streptomycete from one of the isolated from the patient, some of the patients that we may have seen this morning. You would start off by 16S RNA sequencing, getting it into the right genus. Then you would see where within that genus its nearest neighbors were, and then you might do confirmatory tests like looking at the morphology or looking at the chemotaxonomy, or you may have to do DNA pairing, as I'll show in a moment. So this was our initial study. This is a little bit of recent history. We got these strains from Ahmed. We got additional strains from the German culture collection. These had been isolated from mycetoma patients, but quite often a long time ago, and including the type strain. So we did 16S RNA sequencing. When we saw the trees, as I'll show you in a moment, we had to do DNA pairing, and we also did a very thorough phenotypic analysis. And this showed us that people, certainly in this part of the world, and I suspect in other parts of the world as well, when they got a Streptomycete, when they isolated an organism that looked like a Streptomycete, I think it was lumped into Streptomyces somaliensis. I'm not blaming people for doing that. I'm just saying that that was an oversimplification. And when we came to look at these relatively small number of strains, we were able to differentiate a completely new species. Paul showed this slide. We've got the 2 clades with Streptomyces in. The top clade contains the type strain of Streptomyces somaliensis. The bottom clade, where the organisms are all closely related, are the organisms that we now call Sudanensis. And we pinned that down by doing DNA pairing. If you look at the figures in bold under the second group there, you will see they all share similarities over 70%, well over 70% in a number of cases. So I showed you those initial slides to say that 16S RNA sequencing with DNA pairing really is the 2 important techniques to pin down new species. So as far as the genotypic data are concerned, there is no doubt that these organisms are very different. And I think there's about 20 nucleotide differences between organisms in the two groups. That roughly is equivalent to them actually having parted from a common ancestor maybe 10 or 20 million years ago. I don't want to sort of bore you with the phenotypic data, but when you're naming new organisms now, you've got to have your phenotypic data also differentiating between the organisms that you distinguish using these well-known molecular techniques. And the congruence between the phenotypic data and the genotypic data was first class. So we did write a paper describing Streptomyces sudanensis. Now, if I say something, I don't want it to be interpreted as a nasty statement, but it's a little bit disappointing that this work describing this second species really is based upon the collaboration between Ahmed, myself, and the PhD students who did this work. Yet, in most of the papers we've seen over the last couple of days, there's only two people mentioned Streptomyces sudanensis, a prize for who those two people are. And I think this is a pity because we don't know whether Streptomyces sudanensis is the major cause of actinomycete, I can't say it anymore. I've said it so many times, I think my brain's I've not got the capability of saying it yet again. Actinomycetoma. So that is really quite important, and Professor Hassan the other day really underlined that importance. So please don't necessarily take any notice of me, but do take notice of what Professor Hassan said. So these two organisms are very different. So we also were able to say, I'm just showing how we separate some of them here. Let me just see, am I getting, yes, I'm getting ahead of myself, I'm sorry. We got some additional strains, as you can see here. And really the hypothesis was that would these additional strains turn out to be somaliensis, or would they turn out to be sudanensis? You may be surprised that of the small number of strains that have been studied here, but they're quite difficult to work with and quite difficult to grow and quite difficult to isolate. And I think Paul's made a great step forward, finding they actually grow on brain-heart infusion agar. I tried to grow them on about 20 or 30 different media over the years to try and get them to form spores, and failed. So it's a nice example of a bit of luck coming along and solving what looked like a big problem. So we expected that these would be new, and we found, as you can see here, that 3 out of the 4 were new. One of them was another strain of sudanensis. So if we have a league table of sudanensis versus Streptomyces somaliensis, the Streptomyces sudanensis comes out top, rather like Liverpool, and Streptomyces somaliensis comes out bottom, rather like Everton. So we've actually discovered some new organisms, and I should take this point to really apologize to Ahmed, because this work's taken a long time to complete, partly because the student concerned didn't do it properly and we had to repeat it all again, and then I got terribly tied up with Berger's manual, so I wasn't able to push things along, because rewriting the paper actually is quite a big job. But we want to name these species, as you can see. Professor Magoob's agreed. Sobanensis is named after Soba Hospital. And my friend from China, he's just had a genus named after him, so I'd like to relegate him from this and replace him with Professor Hassan's name, if Professor Hassan agrees. That sort of reminds me, we did isolate some new Nocardias, and my PhD student actually said, I would like this Nocardia to be named after me. And I said, well, according to the rules of nomenclature, you're not allowed to be on the paper and have the organism named after you, so you can have a choice. So he chose to be on the paper. But then he said, can I name it after my father? So if you're wondering why Professor Fahal doesn't appear here, he is of course going to be on the paper. This is just to show that one of these organisms, as you can see here, 509, Paul, yes, I did put it in, is a Sudanensis strain. So this is the other strain that's been studied for whole genome sequencing, the type strain of Sudanensis and this second one. And then you can see that these other strains are different in the 16S RNA tree. Similarly here, those organisms at the top are all the Sudanensis strains clustered together on the basis of phenotypic properties, and the other organisms are separated. So we've got a beautiful relationship again between the phenotypic data and the genotypic data. I put these slides in hoping that there'd be at least a couple of vets here, and there are, so I'm very, very pleased. Mohammed Hamid and the group that you can see on the bottom of this in the reference, we showed some years ago that it looked as though Streptomyces were associated with fistulous withers, and we followed this up. This is a very quick summary of this work, using this strategy that I've just outlined. So we did everything properly again. And what was very interesting here was that all of the 7 strains belongs to totally different Streptomyces species. And Mohammed Hamid, who some of us know, he's a very, very careful worker. I'm quite sure these organisms came from the fistulous withers. So it's another example of Streptomyces looking as though it's an etiological reagent. This is the position, these are the nearest neighbors of these strains in the tree. They're pretty well separated, except for the top 2. And these organisms look as though they're a long way from the human pathogens, though I'll come back to that a little bit later on. So, if you can think back to the slide where I showed that there were 8 strains that caused 8 plant pathogens, and then there were our 2 Somaliensis and Sudanensis, our 2 human pathogens, we've now discovered another 10 organisms that belong to new species and seem to be pathogenic. So here in the Sudan we hold the world record for isolating Streptomyces that look as if they're pathogens of either humans or animals. So that, I think, is a step forward, and it does make one think how many more pathogenic Streptomyces there might well be that have gone unrecognized. So very quickly I'd like to move on to Actinomycete, another organism I've been working with for over 40 years. And I don't want to go into a big study here, but broadly speaking, I've plucked a few of our results out, and they indicate much the same as what we've found with the Streptomyces. So these were organisms that we got from the German culture collection carrying the name Pelletieri or Madurae. And in a very big study of the genus Actinomycete and Streptomyces, we looked at these and other pathogens, pathogenic Actinomycete Madura and Actinomycete Pelletieri strains, but I want to sort of simplify it just to make the point. So we took those 5 strains, we studied them thoroughly, and as you can see here, all 5 strains belong to different species. And if you look at the tree, I'm only giving you the tree because I don't want to be too repetitive. These organisms are separated out from one another quite sharply, as you can see. You can get an idea of the relationships between these organisms by the length of the vertical stems. And if we go on to the next slide. So I was prompted to do this because I worked on what were called Nocardia Madura and Nocardia Pelletieri strains many years ago, and I did think there was evidence from the numerical taxonomic data that they were heterogeneous species. So they are heterogeneous species, and the point is, here we've got additional strains that cause this dreadful disease, Actinomycetoma. So what are the outcomes of all of this? Well, in many ways it depends on what people here care to make of it, but the causal agents of Actinomycetoma are grossly underspeciated. I'm quite clear about that. We've got several new species of Streptomyces and Actinomaduras, and I need to put a move on trying to resurrect some of these old data, tidy it up with some more 16S RNA sequencing and so on, describe these new species before I sort of retire from the scene. One of the things that Paul's mentioned, and it intrigued me for years, is that these organisms don't form spores. Have they lost the capacity to form spores? I think we'll find that out from Paul's work fairly early on. I think from that wonderful initial analysis of the 16S RNA whole genome sequence of the type strain of Streptomyces somaliensis, I think it's abundantly clear that these organisms have really adapted to life on human beings, and they show a preference possibly from sweet and cultured people here in the Sudan. So, but also, I mean, I can understand the problems here, that people rely sometimes on the color of the grains and so on for differentiating between organisms. That's what I would call a good artificial classification, but don't take it too seriously. And I think the interesting thing is that when Ahmed and his colleagues isolated what turned out to be Sudanensis, I bet you they had the same type of grain as organisms he isolates that are called Streptomyces somaliensis. So we've got to beware a little bit there. One of the things it's difficult if you're a taxonomist to be proud of anything, because you're not sure if you've done anything useful in all the years you've been studying these organisms. But I'm particularly pleased that the discovery of Sudanensis and showing that Somaliensis was heterogeneous has allowed us to choose good quality strains for the whole genome sequencing. And I think that is, in fact, a big plus. And I think we've got to get sequences now of Nocardia brasiliensis, of Actinomadura maduri, Actinomadura pelletieri. And I think one of the recommendations I would make when we come to recommendations this afternoon is let's get a league table of the strains that we think are worth sequencing, get that onto Wendy's website, and as people get a bit of money, they might actually do some sequencing for us, as Ralph Kirby has done in Taiwan on money from the Taiwanese government. So let's try and get some money from other governments as well, not just Taiwan. And I think the fact that Ralph has done this in Taiwan, and certainly at the behest of Paul and Yola, who we mentioned earlier, I think Ralph worked here in Khartoum for a couple of years, quite a number of years ago. So these networks that Wendy was talking about and talking about developing, I think are a marvelous way forward. So what remains to be done? Quite a lot remains to be done. What are the dominant species that cause mycetomas caused by streptomycetes? Perhaps to a lesser extent the same for Actinomadura. We need to do more epidemiological studies, which is fairly obvious from points that we've had before. And then I return to this point that these organisms that are causing actinomycetoma, from a taxonomic point of view, they are very, very different indeed than the Actinomaduras, the Nocardias, and the Streptomyces. And that makes me feel that we might, rather like in the case of Streptomyces scabies, be having this problem with, now I have problems with this. Can somebody, my eyesight's too bad to sort of get rid of these things with this business. Yeah, thanks, Paul. Yeah, great, thank you.